Hello, good morning, everyone. Thank you for participating in the NIH Metabolomics Interest Group webinar series. Today's webinar from Dr. Pieter Dorrestein will focus on reproducible global chemical analysis of biology by mass spectrometry. Before I introduce our speaker, I'd like to quickly review a few logistics. First, should you experience any technical difficulties, you can contact us through the questions box in the expanded control panel, or by phone or by email. If you need to view live closed captioning, please click that link and it will appear in the chat box. At the end of Dr. Dorrestein's presentation, we will allow time for questions. If at any time, though, during the presentation you have a question, please type it into the question box and I will ask on your behalf during the Q&A session. Basically, as you think of your question, please send it to us so we can get it in the queue and assure that we are able to then be prepared to start the question-answer session on time. All right, so it is my pleasure to introduce Dr. Pieter Dorrestein. Dr. Dorrestein is a professor at the University of California, San Diego, and is trained as a chemist with a focus on understanding how microbes made amino acids, vitamins, and other small molecules such as virulence factors, quorum sensors, and therapeutically valuable natural products. Currently, Dr. Dorrestein is the director of the Collaborative Mass Spectrometry Innovation Center and a co-director at the Institute for Metabolomics Medicine in the Skaggs School of Pharmacy and Pharmaceutical Sciences and the Department of Pharmacology and Pediatrics. Since his arrival to UCSD in 2006, Dr. Dorrestein has been pioneering the development of mass spectrometry methods to study the chemical ecological crosstalk between populations of microorganisms, including host interactions for agricultural, diagnostic, clinical, and therapeutic applications. So we are very excited to have Dr. Dorrestein today with us, and we are going to turn the controls over to him for his presentation.
You can see my screen now?
Yes.
Okay, great. Well, thank you for asking me to give this webinar, and certainly good morning to everyone, at least in California, maybe good evening if you're in Europe or elsewhere in the world. And thank you for the introduction. So what I'm going to try to do today is really highlight where we are with reproducibility and mass spectrometry analysis, where we are, where we want to be, and maybe think about how we might be able to get there where we want to be. I think if you look at mass spectrometry in its current stage, you know, the instruments have gotten so sensitive, the scan rates are so enormously fast. It means that we've entered really, truly an amazing time for understanding biology at the chemical level. Really, we can collect more high-quality data, high-resolution data than ever before. And so if you look at the top 3 lines of this particular image that I show you here, that is really the decreasing costs associated with mass spectrometry analysis. And so this is the cost per data volume that's associated with mass spectrometry. And so this is high resolution, medium resolution, and low resolution, but each one of these different infrastructures, mass spec tools, the cost has come down about 2 orders of magnitude in the last 15 years. What that means is that we actually collect a lot of information. But one of the limitations is that we're not using all this information. But imagine if we could actually use all the information that we can actually detect by mass spectrometry, all the data that we can see, all the information that we can see by a mass spectrometer, if we can actually utilize this. And also if that information could be reused. And so if you look at the red line, this is in comparison to sequencing. There, the cost of sequencing has decreased about 6 to 7 orders of magnitude, and we're really in an exponential decrease in cost again in the last 2 years. And so what the sequencing community has done in comparison to mass spectrometry is they're able to answer really big questions or sometimes even simple questions, but in a very efficient way. So for example, one of the things that the sequencing community is able to do is to ask the question that if you have a sequence, you can say, ask the question is, where's this sequence been found? Where are the analogs found? What are the analogs that are present, that are known in the public domain? And then what are possible functions associated with these sequences? And the reason why we're able to answer these questions in sequencing is because the community has basically created a database with genomes, metagenomes, genes, and when they characterize individual gene functions, those annotations are retained as the knowledge base. And then they build retrieval infrastructures on top of it, so that as soon as you have a sequence, you can do a multiple alignment search such as BLAST and very quickly retrieve this information out of it. Now, if we contrast this to where metabolomics is or mass spectrometry is at the moment, and you ask the same question, where is this molecule found? Are there any analogs found associated with that molecule? And what are the possible functions associated with that molecule? You'll find out that this is a very difficult problem. And the reason is we may have some data that's stored in the public domain. And so projects in metabolites, metabolomics workbench, but the majority of mass spectrometry data is not stored in the public domain. Then you have reference spectra, which are great databases, and on the right side of the screen you'll see a number of these. But now let's explore how we tackle this problem. If we ask the question, okay, where's this molecule found? In what projects? Well, you have to download all the projects. You have to download all the reference data sets. You might have to develop some scripts or new algorithms first to convert all the data in a uniform format, and then secondly, to be able to do the searches. And so to answer a question, where's this molecule been found before in all the metabolomic and mass spectrometry data, it turns out that this is actually a multi-year problem that is really hard to do, and that is very much in contrast to sequencing. And that is because we don't have a centralized retrieval infrastructure, and in reality, in many ways, there's not enough data in the public domain. But what might it look like if we tackle this problem in mass spectrometry? And maybe we should look at some infrastructures that have been extremely successful in being able to tackle a problem like that. And that is a lot of our social infrastructures, or even the Google search engines. Facebook, Yelp, Reddit, Wikipedia. And the way they started was they basically started as a database. Then they figured out ways to capture community knowledge. Then they figured out ways to build relational algorithms so they can mine the knowledge associated with the database. And then they have retrieval infrastructure so it becomes useful for the end user. And I think over the next few years, mass spectrometry is going to get basically as good as Facebook or Google and as easy to use. Now, the community has realized that we need databases. And I just showed you in a previous slide, there's a variety of different databases that exist. But what is limiting now is to actually begin to mine the databases and the knowledge associated with it. And so first, what we basically need is a set of relational algorithms. Now, before BLAST was developed in sequencing, there were probably thousands of algorithms that were developed to build relationships. And ultimately, BLAST was one of the tools, one of the algorithms that turned out to be most successful, even though other relational algorithms are still being used. The mass spectrometry community started to build these type of relational algorithms. We released one in 2012, which is called molecular networking. But there are other tools, such as the ones that are created by Emmanuel Garrel that is based on information theory. There's also the topic modeling released by Justin van der Hoofd or Simon Rogers. These are basically tools that build relationships among the data at the single molecule level. And these are really, really powerful. But I'm going to focus on molecular networking because that's the infrastructure, the relational algorithm that we have developed. What molecular networking allows one to do is basically create a map of all chemical space that was detected by mass spectrometry. And it does so based on relationships between the spectra. And the way I think about relationships between the spectra is that this is a relationship among the molecules that we could detect by mass spectrometry. Now, for the people who are not exposed to molecular networking, I'm going to briefly introduce how we do this. So one thing you have to think about, if you have two identical molecules or two related molecules that are structurally similar, not identical, but related to each other, if you subject those to fragmentation, their resulting fragmentation patterns should look very similar. And in most cases, it really does. And we can use that principle to look for similarity among the MS2 spectra that we're able to detect. And so we align these different spectra. And then instead of representing these as spectra now, we represent these as nodes. And the way I think of these nodes is that these are the unique ions that we were able to observe by mass spectrometry, and that translates to the underlying molecules. And so now we get these groups of molecules, and then we connect these groups with nodes. And we often use the thickness of the edges to define the degree of similarity. Now, but in a given untargeted metabolomics experiment, we don't just have 7 spectra. No, sometimes we have millions of spectra that are associated with it. And so it becomes much more complex. And so you create this molecular network, and basically the way I think about this molecular network is that this is a map of chemical space that we were able to detect in a given untargeted metabolomics experiment. But then what is nice is that we can take reference spectra and we can begin to populate this particular map. And so what a typical project when people do untargeted metabolomics does when they publish is they only publish these annotations and they ignore everything. In general, they ignore everything else. And this is unfortunate. But I'm going to zoom in on one of these subclusters within this molecular network. And we often refer to these as molecular families because, in principle, the chemistry should be fairly similar. And so here's an example of one of such networks, and that is shown right here. And this was a molecular family that we showed in this particular paper that we recently published. But this was a molecular family that came out of a molecular network where we looked at hundreds of Pseudomonas strains from different origins. They came from humans, they came from mosquito guts, they came from plants, and we wanted to see what are the chemical relationships or the molecular relationships among these different data sets. And so in this particular case, we were able to match to a reference spectra of viscosamide, this particular node, and then this was produced by one specific organism. But then there were other organisms that made Pseudomonas strains that made related molecules such as viscosin. And you can see that it's just a single amino acid substitution. Then we have, oops. Sorry, I went a bit fast on the key. And then we have another organism produced macetonolide, and it's just a single amino acid substitution. Then we have an unknown molecule. This was just an addition of a single amino acid. Then we have orphamide that is shown right here. We have tensin. And then, again, we have some unknown molecules that are associated with it. But essentially, what molecular networking allows you to do is begin to propagate a lot of these annotations. And so, let's say one of these molecules was a drug and you wanted to study drug metabolism, a lot of the metabolites will co-network with the original parent drug, for example. Now, when we started molecular networking, we actually did this with a very small number of data sets. So let's say 1 to 10 data sets, LC-MS or atmospheric ionization methods. And we did this on one computer with 4 gigabytes of RAM. But then we decided, okay, we wanted to do more data sets, and so we invested $5,000 to buy an expensive computer at the time with 64 gigabyte of RAM, and we were able to compare 100 data sets. But as we started to want to compare more and more data sets, we ended up moving to a 300-node cluster. That allowed us to compare 1,000 data sets. And then in 2014, we wanted to do thousands of data sets. We moved to a supercomputer with thousands of nodes. And then even then, still 3,000 data sets took 2+ days. Now, ultimately, we wanted to compare hundreds of thousands of data sets. And we really thought we needed to be able to take over the NSA computer, but in reality, we needed to become smarter. But what when we published the paper on molecular networking, there was a lot of enthusiasm for it. And a lot of people wanted to perform molecular networks. And so we made the code available to the public. But every single person that downloaded the code, they couldn't get it to work. Basically, what it took was, it took a computer scientist, a very well-versed computer scientist and with a lot of experience to be able to install this particular code. So what we did instead is we set up a remote computer, and we sent people, we provided them with remote access to this computer, and we sent them 35 pages of instructions to create a molecular network. Now this was successful. There's a Nature paper where they showed that new environmental taxon had a distinct metabolic repertoire. It was used to show that Streptomyces silicola makes an unpredicted natural product suite. It was used to find the molecule silibactin, which is a molecule that is thought to be responsible for initiation of colorectal cancer. The genes were known for a decade, but the molecule had eluded detection, and molecular networking enabled them to find this particular molecule. But it turns out that the needs expanded beyond our capacity. And so this is why we created a web-based infrastructure. And we published on this in 2016, the end of 2016, and it was really a worldwide effort. And what is GMPS? So this is the Global Natural and Social Molecular Networking, but it's basically a database, it's a knowledge base, it uses the relational algorithm, in this case molecular networking, but it also has the retrieval infrastructure. It's the database, knowledge base, and that retrieval infrastructure. And in our case, this infrastructure really was born out of need. It was really tiring for me with large mass spectrometry projects, and we wanted to share data with others, that we had to send hard drives to other laboratories. And then, in terms of the knowledge, if I had a graduate student that graduated after 4 years, they spent 4 years on these particular data sets. They've gained a lot of knowledge, but as soon as they graduate, some of that data may be, or some of the knowledge may be embedded in papers. But in my opinion, because there's 2.5 million papers published each year, as soon as the paper is published, that knowledge is lost to the community unless it's in a digital format. And so when people would graduate and somebody else would pick up on that same data set, they would actually have to start from scratch. So we wanted to find a way to capture that knowledge. And that's really, in many ways, the database sharing ability and knowledge capture is what GMPS is really about. Now, right now, we actually have a lot of users. We have 136 countries that are using it, 25,000 users worldwide that are using this particular infrastructure. And you can see how much it's being used. What I like about the GMPS infrastructure is that different communities are beginning to speak the same language. So somebody in forensic may make an annotation, and those annotations may actually be useful for people who are studying dissolved organic matter, or they may be useful for people who are studying exposomics in humans or in precision medicine. And so the reason why this is possible is because the public data is growing very significantly within this infrastructure. And because the public data is growing, we can now begin to cross-correlate the information that is collected by the different communities. And so one of the things that is really growing within the GMPS infrastructure is the knowledge base. The knowledge base are the reference, basically the reference spectra associated with spectra within the GMPS infrastructure. The first thing that we did is to build this knowledge base is actually we connected up with all the public repositories that existed. So we moved all the reference spectra that are in the public domain into GMPS, and we even share directly all our reference spectra with other resources such as Mass Bank North America. But what that means is that if data is available publicly, we can search against NIST, ReSpect, HMDB, Japan, Mass Bank North America, Mass Bank Europe, etc. But then the question is, how do we capture the community knowledge? So this was already knowledge that existed in the public domain, but how do we capture the knowledge by the GMPS community? And there's a variety of strategies that we've implemented to capture knowledge within this particular infrastructure. One of those ways is that you can run an MSMS spectra of a pure standard and add it to the GMPS library. The other thing that we've enabled is, because a lot of molecules you cannot commercially purchase or would be difficult to synthesize, but yet we see them in our LCMS runs or atmospheric ionization experiments, you can provide a single scan number and actually annotate that particular single scan number, what it might be. You can also do batch uploads, so if you have a lot of reference compounds, and below here are some examples of these libraries that have been uploaded in batch formats, you can do that as well. But also, as a part of the GMPS workflow, we've just routinely provided the ability to add to library. And basically, this is an annotation that is provided that then now becomes a part of the community. And so annotations such as sodium formate clusters, polymeric compounds, multidimers, heterodimers, chimeric spectra, these are all now part of the GMPS annotation workflow, and they are now shared with the community. When you make an annotation, it actually becomes a part of almost like a wiki page associated for every spectrum that's within the GMPS platform. Right now it's about 2.5 billion spectra or so that are within the GMPS platform, but essentially it has an annotation history. It has comments associated with these annotations if the community has provided this. And so this is really the knowledge base associated with GMPS. Now the knowledge base is actually growing fairly with a reasonable pace within the GMPS infrastructure, and I've just shown the growth for it for the last 2 years, where the GMPS community has totally provided 70,000 reference spectra to be and this is available to the entire the world community. Now, when you look at that inset, what I show is just a gradual increase of annotations within the GMPS infrastructure, or a reference spectra, I should say. What I like about this line is that it's often just single contribution. So a single spectrum might be annotated by a person, and that, we capture this, and now this becomes community knowledge. And then you can see the enormous growth that we've seen since we've published a GMPS paper, and it's very clear that people are starting to contribute reference spectra to build the public knowledge. Now, one of the questions, of course, now we have these large data sets in the public domain. There's about 560 projects that people have made publicly available. This is in contrast to the 910 projects that are totally available in the public domain, where you have raw data, if you include Metabolomics Workbench and metabolites. But the question is, how much of the data that people upload in the public domain can be annotated with these reference libraries, with the knowledge that's accumulated in the public domain? Now, if you have well-studied organisms or a relatively simple system, such as cultured E. coli or cultured human cells, we can right now annotate 8 to 10% of the data when you do an untargeted metabolomics experiment, at least in my hands. If you go to well-studied systems such as human samples, such as plasma or urine, we can get up to about 5% of the data we can annotate. But on average, within the GMPS infrastructure, we can only annotate 2% of the data. There are some biological systems where we get almost no annotations whatsoever. And that simply indicates that the reference libraries are really biased towards what we commonly study. And in many ways, these numbers are actually going to become worse before they become better. And that's really because for every order of magnitude increase of sensitivity of instrumentations, the number of unknowns are going to double. And this is very similar to what the sequencing community went through. Now, if you looked at a genome about a decade ago or 15 years ago, most of the open reading frames that were present in the genomes would be unannotated. But now, a large proportion of the genes are annotated. And so we're actually, in the sequencing community, they're starting to decrease the number of unknown open reading frames. And we'll get to that in mass spectrometry as well, but I think these numbers are going to become worse before they're going to become better. But that's only a good thing. It means that the instrumentation becomes so powerful. Now, one of the unique things that we do within GMPS is that we've created this concept of living data. What living data means is that if you make your data project public, so let's say in Hawaii somebody uploaded 16,000 LCMS runs. There's other people that have uploaded 20,000 LCMS runs within our infrastructure, and they made them public. But they make annotations. In fact, anybody in your lab can make annotations to that data set, or anybody in the world can make annotations to that particular data set. But let's say you make an annotation to a specific spectrum. If that spectrum matches to somebody else's data that is public, that person will get an email, or any subscriber to that particular data set, they will get an email. And this is true for any other annotations that are matched behind the scenes, because behind the scene we cross-correlate and basically create one super molecular network. And what that means is that we really get this cross-learning about molecular information, and we disseminate the best knowledge that currently exists in the public domain associated with untargeted metabolomics data. And so when you get an email, it will look something like this. You can click on the hyperlinks, and you can look at all the annotation. You can download the data tables, et cetera, et cetera. And so what that means is that maybe this year you upload a data set, and maybe 10 years from now, you will get an email saying, hey, you have new annotations, and maybe that annotation may make a change in understanding a very fundamental hypothesis you've always had. And to me, that's really exciting, and it changes the way we actually interact with data. But of course, when you rely on automated tools, mistakes are going to happen. It could be that there's an incorrect annotation, or a annotation that needs to be refined. And so there's a couple of solutions to this, so that while mistakes will be made, it still enables us to provide higher confidence in the annotations that we have. One of those is that we provide the ability to actually take advantage of the community in terms of collective improvement of annotations of the information that's within GMPS. So let's say somebody said, I have an MSMS spectra and this is a sugar. This is great. That's a great annotation because a biologist may care about this. But somebody else may know that this is not just a sugar. I know it's erythromycin. It's basically this antibiotic. And here's the supporting information of that. And let me fix that for you. It may even be possible that we have two different molecules that almost have identical MSMS spectra. Well, we provide both of those annotations. And the key part is we remember every single annotation that is made so that the end user can decide what annotation is most appropriate for them. So this is how we take advantage of the community. We further take advantage of the community in the ways that other social infrastructures do this. And that is through a star rating. So one of the things that we don't have yet, and I will get to that in a second, we don't have a good false discovery rate associated with annotations in metabolomics data. And so because of that, we decided we need to figure out a way to provide some sort of quality estimation associated with the matches that we do provide back to the community. And we again leverage the community by using these star ratings. And that is shown right here. So we have this concept of living data where we periodically annotate information. And here we're looking at one data set, and this data set has 6 subscribers. When it was uploaded, it had 8 annotations, and within a few months it grew to 49 annotations. You can look at this, and then you can look at the star ratings. And basically, now you as an end user can decide whether you trust these annotations or not. So for example, 1 star would be an incorrect annotation, 2 star where we don't have enough information to tell if it's correct or not, 3 star that it's an isomer or possibly correct, and then 4 star would be correct annotation. And then this plus button that you see on the right there adjacent to the stars it basically allows you to provide comments associated with this particular annotations. And all this information goes back into that wiki page associated with the spectrum. What that means is that people can have a dialogue in terms of the quality of these different annotations and have a discussion on this. Now, I did mention that we did not have a false discovery rate that's routinely implemented for annotations of metabolomics data. In fact, up until recently, we did not have this. But I teamed up with Sebastian Booker in Jena, and he basically used his decoy data, or his spectral tree approach, to generate a decoy database. And the way this works is that if you have a library, a reference library, you can think of the MSMS spectra as a tree. Each fragment I is a branch of the tree. But one of the things that you can do if you want to be able to calculate a false discovery rate, you need to have a decoy database that actually looks like the library. The way this was done here is that we break these branches apart and then regraph these branches onto the tree itself, and that essentially creates our decoy database. And so now when you do searches, you search both against the library as well as the decoy library, and you simply count the frequency with which it matches to the decoy library to estimate a false discovery rate. And this is important because it allows you to start defining what are good instrument parameters to use for a given data set. And it turns out that every project has different requirements in terms of scoring criteria that you use to get a proper false discovery rate, such as a 1% false discovery rate, if that's what you choose. So for example, if we have 3 minimum match peaks, your scoring function needs to be much higher. And one of the things that you will notice is that it's project specific. So if we look at the frequency here, that's the frequency for a given data set. So this is 70 different projects within GMPS that were analyzed. But any given data set, they require different scoring functions to be able to get acceptable false discovery rates. Whereas if you take 8 ions, you can be much more of a, if you have 8 ions that you can match within this using an FDR calculation, now you can relax your scoring criteria much more because you have much better scoring matches due to the increased number of peaks. And so the key message here is really that we need to adjust the scoring parameters on a per project basis. And so what is GMPS enabling that was not possible before? Well, one of those things is it allows you to explore the molecules that are present. So for example, let's say you're interested in the molecule cocamiddyl propylbetaine. This is a common ingredient that are found in shampoos and soaps and such. But you want to know where this molecule has been seen before. Now, it turns out that this is a molecule that's very commonly a contaminant in mass spectrometry. But let's say you were interested in this. Well, with Molecular Explorer, we can look for this molecule, and then very quickly, we can find all the data sets on the right where this molecule has been observed before. We can also do single spectrum searches. Let's say you're interested in one specific spectrum, but you don't know what it is. You can actually add that spectrum to in the GMPS infrastructure, even though it's still a test version, but you can find all the data sets that have that particular spectrum. And this was not possible before, and turns out that this is often very useful. The other thing that you can do is you can compare and contrast data sets at the MSMS level. So here's an example where the Jensen lab was interested in drug discovery from actinomycetes. And they basically had a culture collection of 1,000 actinomycetes that they subjected to untargeted metabolomics analysis. And one of the things that you will see is that the number of unique MSMS spectra that are collected as the number of strains grows, it eventually reaches saturation. And so if he were to continue to collect actinomycetes from these same locations, it basically wouldn't give him an increased diversity of molecules for his drug discovery program. Whereas if you compare that in contrast to another actinomycete library, the Linnington library, that's another 300 or 400 strains, and you compare that to the Jensen library, now all of a sudden you see that there's certainly an additional number of unique MS/MS spectra, and therefore an additional potential for discovery of additional molecules in these drug discovery programs. And really for them, this was informing them is that, yeah, we need to start collecting actinomyces in different locations so that we can increase the number of unique molecules that we encounter in our drug discovery programs. The other thing that GMPS is able to do is it allows for export into Cytoscape or QIIME or Cheetah. Cytoscape is a tool, and I showed some molecular networks that were built with Cytoscape, but essentially it's a tool that the NIH invested at least $40 million into. But it allows you to get an overall overview of the types of molecules that are present within this infrastructure. But there are other great infrastructures that exist in the public domain. QIIME and Cheetah, even though they're designed for microbial ecology, they turn out to be very useful for mass spectrometry analysis as well. So QIIME is basically a statistical package analysis and visualization tool that has been cited 7,000 times over the last 5 years. But essentially, it allows you to do multivariable statistics such as Bray-Curtis, random forest, etc., and then visualize that with all the metadata that exists. And I'm just showing you an example of how we've exported some data from the GMPS platform into QIIME at this point. And this is a study where we basically took 4 germ-free mice, 4 microbial colonized mice, and we asked the question, how does microbial colonization impact the chemistry associated with the different organs in these particular mice? So we took 29 organs. We have 3 to 4 samples per organ of 8 mice. And then we did a Bray-Curtis analysis within the QIIME infrastructure. And then what we can then do, if you have metadata, you can really on the fly look at the metadata associated with it. So here we're looking at distribution within organs. Here the wells where these samples were collected, or whether the sample were germ-free mice or microbial colonized samples. And so there we can start to see really the differences and the impact of microbes on the different organs and such. The other aspects that it is able to do, you can look at different PC axes or PICO-A axes associated with it, and sometimes that allows you to find different drivers associated with the biological differences that we do look at. But what I really like about this infrastructure is that we essentially can provide a hyperlink in publications or 2 folders in publications so that people can re-look at your data, but also understand how the data was analyzed. And this gets us to data analysis reproducibility, which I think is really an important thing that we need to consider in untargeted metabolomics. So let's say we have the exact same data sets, and we give it to a number of different people, and we give them the same informatics tools, and we have the same question. The question I have is, will we actually get the exact same answer out of it? And the answer is simply, in my opinion, no. And I think this is probably true. I can give any of my data sets to any of you that are listening. You're not going to get the same answers as I do. Not the same answers in terms of annotations, not the same answers in terms of what are the most important molecules that are differentiated if you have 2 different cohorts, etc. So this is a real bottleneck, and this is not just a problem in the mass spec community, it's also a problem in the sequencing community, and it's something that the NIH and other funding agencies are very interested in solving. Now, the way we deal with this within GMPS is we at least want to be able to make it possible for the community to look and repeat the exact same analysis so that you get the same results. One of those ways we do this within the GMPS infrastructure is that we remember every job that is done. You know, maybe the way to think about this, Google remembers every single search you do based on IP addresses and such. we do essentially the same thing, that we remember every analysis job that you have done. Now, of course, your jobs are in your own user space. When you log in, only your jobs show up. And so this is my login page. I've done a total of 317 searches myself within our GMPS infrastructure. This does not count the workshops that I did. But essentially, we have every analysis job that I've done. This could be molecular networking or library searches or any of the other tools that we have, such as CSI Finger ID or MetFrag searches that are in silico library searches that we also have. So what is nice about this is that you can now click on any of these done links. What that will do is it opens up a link in a web browser, and you can share those links in papers. And so here, for example, we've shared one of those analysis runs for molecular networking results within our paper. You can click on that, and it will bring you to this window shown right here, where you can look at all the compounds. You can export the data for Cytoscape visualization. You can look at a variety of different parameters associated with it. But the other thing that you can do, and any user can do this, you can clone the job. It recalls the data set, all the data sets, even if you took 10 projects out of the public domain, it recalls all of those, and it recalls the reference library, and you can reanalyze the information in a very efficient manner with the same parameters that I used. And then, if you want to make slight changes to those parameters, you can do that easily as well when you hit the clone job. And so I think, GMPS is one infrastructure that is trying to tackle the data reproducibility problems, but there's certainly many other infrastructures that are tackling this particular problem, and just a subset of these are shown right here. But what I hope that I see will happen in the future is that these different infrastructures will start to communicate with each other, because each of these different infrastructures really serve different communities, serve different functions, but one function may be useful for another infrastructure. But what might such a connection look like? And this is shown in this particular case, where we're connecting different resources for reproducible data analysis. And here I share a link in a discussion with metabolites and ILI and GMPS to show how we can link different data sets to different analysis infrastructures. And when you click on this hyperlink, what it actually does, it brings you to a analysis tool called ILI in GitHub. It draws the three-dimensional file from a separate website. Then it draws the coordinates and the data from GMPS. And now you can actually look at the data in the way I looked at it in a reproducible fashion, and anybody could scroll through this. And I think this is one of the ways how we may be able to link all the different infrastructures for reproducible data analysis. So finally, I want to leave you for the last 2 or 3 minutes highlighting an example of the use of GMPS and the scale with which it can be done. So this is a project that is done by my postdoc, Alan Jarmusch. He's been interested in basically using mass spectrometry for clinical diagnostics. And one of the areas that he's interested in is using fecal samples as a way to do clinical diagnostics. And there's a long history of using fecal samples for clinical analysis. And basically, the way it's used right now in the clinic is through looking at the consistency of fecal matter or at the color of fecal matter. And these actually give you insight in terms of the well-being of the person that it came from. If you're outside of this color range, it is recommended that you go and see a physician. Now, Alan is a really talented analytical chemist. He came from Graham Cooks' lab, and he felt, just by eyeballing it is not analytically very satisfying, and certainly for his perspective, he has a hard time thinking about it in the context. How can we apply this routinely in the clinic? And so the first thing that he wanted to do is he wanted to understand basically what's the typical molecular makeup of fecal samples. And he also is interested if there are geographic distributions throughout the world associated with the molecules of these fecal samples. Now, maybe a practical question is, how do you get a large number of representative fecal samples? In reality, grad students or postdocs are not representative. They would certainly be willing to sit over a cup and provide you material, but it certainly wouldn't be representative of the population. And so how do you get a large number of representative fecal samples? One of such ways is through crowdsourcing. So we're part of a crowdsourcing project called American Gut. And basically what this crowdsourcing project does is you can basically pay $100, then we will send you a sample kit, and we will look at these samples for microbiome sequencing, and we're starting to do the metabolomics on this. And this is where Alan basically got 2,400 samples, and he did a standard extraction, resuspension, and then LC-MS analysis. And now we had to deal with 2,400 data sets. And if you look at the number of MS-MS spectra within this particular data set, it comes to 2.8 million MS-MS spectra. And for most people, they don't have the capacity to deal with this, but GMPS enables us to begin dealing with this volume of data and in a reproducible fashion. So one of the things that he decided to do was, well, I only want spectra that are information rich. And so he said, I'm only going to keep spectra for which we have 6 product ions. And that reduced the complexity or the number of spectra to 2.3 million. Then he said, well, I want to make sure that the spectra that we're going to consider and that we're going to pay attention to are observed fairly frequently. And so he said, I'm only going to keep spectra that we've seen at least 5 times. And this is all really easily done within the GMPS infrastructure. And this reduces the complexity to about 31,000 unique spectra, because all the spectra that we've seen 5 or more times, they are merged together if they are unique spectra. And so this reduced the complexity to 31,000, and that's a manageable number. And with that, he was able to create this molecular network and then add metadata to this. The metadata that he has added to this particular picture was the molecules that were unannotated were shown in blue. The molecules for which we had reference spectra are in the public domain are annotated in red. And then the molecules that were observed very frequently, they are shown as large nodes. So if they are seen in 24,000 or 2,400 times in each one of the samples, those are the largest nodes. And whereas the spectra that are not as frequently encountered, they are the smaller nodes. There's a couple of things that you will notice. For one, it's only about 5% of the spectra for which we have annotations for. And then also a large number of molecules that we do detect, we don't have reference spectra for. And so it again shows a bit of a bias that are present within the reference libraries. Now I'm just going to zoom in on one molecular family that had an annotation within this molecular networking, and that is the papain molecular network. So this large triangle shown in red, where we had reference spectra in both mass bank and NIST 2014, and this was a perfect match to this particular molecule. And then adjacent to this is we had a match that was 14 Dalton separated, and that was a match to somebody from the GMPS community had contributed this particular molecule.
And you can see the difference between these two annotated molecules is 14 Dalton, and that really resides from this, the part of the molecule on the left side where we have a 6-membered ring versus a 5-membered ring. And then we have additional nodes that are not annotated in blue, but we can begin to propagate a lot of these annotations by simply looking at the mass shifts that are associated with it, and then careful inspection of the MSMS fragmentation patterns, and actually see the relationships between the annotated molecules and unannotated molecules. But this is one nice way to begin propagating some of the annotations. Now, because we also have the coordinates associated with where these samples come from, we can also inform ourselves of these particular molecules and the distribution of these particular molecules. And so the way this is done is we take the coordinates, GPS coordinates associated with these samples, and we actually map this into ILI again. And so here we just show by pairing molecule, and we can see that it's distributed in 14% of all the samples, and we can see this in Australia, United Kingdom, as well as the United States. We can also look at the number of drugs that people are taking, and we can provide statistics for it. And in this case, we clearly show that the number of drugs per subject in the United States is much higher than other parts in the world. We can look at individual drugs as well, and here's an example of scopolamine. We can look at the distribution, and we can only find it in the UK. Now, it turns out that this molecule is approved as an over-the-counter drug in the UK. It's approved in Australia as a prescription drug and not approved in the United States, and that's probably the reason why you only see it in the UK. Now, this one I find very interesting. We detect THC in fecal samples, but we only find it in the United States samples and not in the European samples or the Australian samples. And I think we can all come up with interesting hypotheses of why that might be, and that may be a better discussion to have over a beer or a drink, because there are some very interesting correlations there. But we can also export this information very quickly into Cheetah, and now we can do a global scale analysis and compare it with the data that, with the metadata that we do have in a very efficient fashion. So 151 of the data sets that were collected came from fecal samples associated with intensive care unit. And you can clearly see in this multivariable space that people in the intensive care unit have very different fecal samples than the general average American gut sample. And so I think ultimately we'll start to add other clinical cohorts to this particular information. But that's why public data is so important, because now we can compare these kind of data sets. But what I find even more important is that for all the analysis that I've just shown you, the molecular networks, the global maps, the PQA for this project, we could provide links so that anybody can essentially reproduce the analysis that I did. And that's the first step in the first hurdle that we need to overcome as a community to be able to promote data reproducibility. Ultimately, if you want to be a part of this particular map, you're certainly welcome to go to the American Gut website, and there's instructions on how to participate in these particular efforts. But I want to leave you with a couple of take-home messages. And I think over the next few years, and certainly it will be faster if the funding bodies realize, provide funding at the scale as, let's say, the Human Microbiome Project, which was $174 million. The reality will be that untargeted mass spectrometry can be as efficient as Google or Facebook type analysis. But in the meantime, to enable this capacity in the future, we need to share raw data and not data tables. There's a discussion in the field that data tables should be shared and not raw data. I disagree with this. Data tables, you can no longer inspect the raw data if mistakes were made. And the comparison to this is the protein data bank. in the protein data bank, they opted a long time ago initially that only coordinates of crystal structures were needed to be deposited so that people can look at these crystal structures. And what that meant was that the raw data was not available for those older crystal structures. But over the last decade, the ability to analyze raw data for crystal structures have improved so significantly that they could have gone back to the old data and actually improved the quality of structures that we can obtain from the raw data. The same is true with metabolomic data. So feature finding, for example, I would argue that right now is still an art and not really a great science. Different feature finding tools will give you slightly different information. And so having the raw data allows you to, in the future, update, let's say, feature tables. It also allows you to reanalyze the reference libraries. We should also really make sure that we share codes, scripts, and algorithms of any analysis that we do, because this is how we make sure that the data in papers are reproducible. We should also try to really publish so that anyone can, so that anybody who takes your data should be able to get the same results with your data and if they use the same tools. And then finally, I really think this is really important. We need to continue to develop new and innovative thinking in terms of development of relational algorithms. BLAST in sequencing was not the first algorithm. It wasn't the second algorithm. No, it was probably the 1000th algorithm that was developed that became the most popular algorithm. But we need to use these relational algorithms at scale. And then finally, there's a lot of people that I need to thank. This was the initial community, GMPS community, that provided data, that provided feedback, that provided community knowledge. And I'm really grateful for them. And then finally, I need to thank friends, coworkers, and support that allow us to do the research. First of all, the Bandera lab, without him, a lot of the molecular networking and GMPS would not exist. Theodor Alexandrov, the development of ILI, would not exist without him. He's a really fantastic person in terms of data visualization. The Night Lab for all the sequencing and our collaboration, especially in terms of American Gut. And then finally, I want to thank the people who supported the work. And I will take any questions that people might have.
Great, excellent. Thank you for an excellent talk. Very interesting, and we do have some questions. I know we're kind of at 11 o'clock, but we did receive 5 questions. So those of you that are interested in hearing what the answers are, I am going to try to ask those 5 questions, and hopefully we won't go too much past the hour. But the first question, is there a way to include the publication references into this database, allowing us to identify the sources of these NPs?
Yes, so you can update any public data sets afterwards with the publication itself. Any data sets that is in GMPS, you can actually, there's an accession code that we provide, an MSV number, and you can put those into publications. So both of these should be linked. So you can indeed do that.
Excellent, thank you. The next question is, can investigators using GNPS summarize the findings from can it be used to summarize the findings from metabolic findings broadly, or to help understand the meaning of a metabolite in epidemiologic studies? So essentially, can you search the data to determine which metabolites are produced by bacteria?
So yes and no. It's not that straightforward yet. We're building the infrastructure to do so. But basically, one way that you can do is you can, if you have, let's say, a data set such as American Gut, you can search the microbial data sets and at least get the reference. So let's say you have a spectrum that is found in American Gut, and then a spectrum that is found in that particular bacterial data set. You can at least say that that spectrum or that molecule that was detected is found in that particular bacterium as well. And so you can do that with all sorts of bacteria. And so one of the things that we're beginning to do now, we're beginning to create about several thousand data sets where we have a genome sequence as well as metabolomics data sets, so that we can basically have a reference data set that we can search against. So we have an alpha version of a tool like this running, and I'm happy to communicate with anybody on this offline. They can send me an email. But that's exactly where the community is going. And I suspect within the next 2 years, there will be decent tools that allow you to do this in a routine fashion, but it's still a hard scientific problem at this point.
Great, thank you. The next question is, what kind of strategies are you planning to get the fecal metabolome better annotated?
So there's a couple of avenues that you can take. One of those is indeed reference data sets. So not just bacteria we're creating reference data sets for, but we're also creating reference data sets for food. So we have several thousand LCMS runs that are from different food samples that are going into GMPS soon. And these become reference data sets because a lot of molecules we see in fecal samples come from our diet, which makes sense. Then there are a lot of molecules that come from microbes, and so again, reference data sets. And so the way the annotations help is that even though we don't have a molecular annotation, we at least have an annotation of where these molecules might be coming from. Then to provide annotations, we rely on the community. So as I highlighted throughout the talk of the strategies, which the way we capture community knowledge. Also propagation, using molecular networking, we can propagate one molecule to another, and we need to capture that information. The other aspect of it is the in silico methods for annotating spectra are becoming better and better. Now, they're not perfect yet, but certainly if you would have talked to me 3 years ago, I would not think in silico spectra or in silico approaches were reasonable, because 5 years ago or 3 years ago, the CASME challenge showed that only about 5% of the annotations were actually correct. Now in 2016, that number has jumped to 50% of the annotations were correct. And so the tools are becoming better. So one of the first things that we're doing is we're actually building or taking some of these in silico methods, such as CSI, Finger ID, MetFrag, and we're providing those annotations to the communities as well. And again, there we're going to rely on the community to provide us with whether they believe those annotations are correct or not, and we'll capture that knowledge. And so this is one of the strategies forward of how we're going to improve a lot of these annotations. There will always be molecules that we cannot annotate, and for those we have to isolate them and do NMR structure elucidation.
Great, thank you so much. The next question is, how do you export the GNPS data sets into Cheetah?
How do you export? So there's when you do an analysis run, there's basically a feature table that is on the bottom of the link in your analysis run, and you basically create a BioM format, and that in turn is what is imported into Cheetah. So we have workshops on this periodically, and we're happy to provide. People can email me, and we can tell them when these workshops are.
Oh, great! That's fantastic. There's one last question. We're going to take the time to answer it, and I appreciate everybody hanging in there for a little bit longer, past 11. But are there databases that summarize the results of published results from studies? Is there any effort to use machine learning to summarize these findings from the published metabolomics papers?
There are some machine learning approaches that people have been using within a given project. I have not seen machine learning happening at the global scale, but this is exactly you know, the time is right for that to start happening. And that's exactly, you know, that's why it's so important to have the data in the public domain, annotations in the public domain, so that we can begin to do machine learning, basically.
Great, excellent. So those were all of our questions that came in for today, and I would like to thank you all for your attention and active participation in today's NIH Metabolomics Interest Group webinar. And we would also like to take this opportunity to thank our presenter, Dr. Pieter Dorrestein, for an excellent presentation. I thought it was really informative and just fantastic today. So thank you, Dr. Dorrestein, for your time that you spent with us today sharing about GMPS. And we welcome your feedback to inform our future webinars. So if you have any, please let us know. And having said that, that brings us to the end of this webinar. Thank you all for attending, and hopefully you will join us for our next webinar. Thank you, and have a great day.
Thanks.
We need